Hello. Let's wait for some people to log in. I've only got one student so far. Here they go. Yours may have been slow. All right, after a minute, I will start. Looks like we're not going to get very many people logging in for some reason. We did do the lab one, right? Let me go to my announcements section. Okay, are there any questions? If not, let's go ahead and begin. So the lab will be from 6:30 to 8:20, and I will be here from 6:30 until 8 o'clock generally. For part of this time, I will talk about what we're going to be doing. And for part of this time, I will just be here to answer any questions that you're doing with the lab. Any questions about any of that? So the lab is going to be open and online. And I will be here to answer any questions if you have. I'm here for you. All right, let's go ahead and get started. I'm going to briefly just talk a little bit about the syllabus, what we didn't cover in the lecture, because this is the lab. I didn't talk about the lab in the lecture. There's only a few things in the syllabus. I'm not going to go over the grade points. So if you're interested in that for the lab, go ahead and take a look at that. I've already gone over the grades and the points. You're not seeing the screen yet, are you? Let me turn on the screen. Is that down? Can somebody confirm that you're hearing me okay?
Yes.
Yes.
I can hear you. I just can't see your screen.
Yeah, I'm working on that. For some reason, the lecture is only 30 or 50% now done and converting the lecture into meeting notes. Let me see. Share screen. I don't think we'll use a video, but I'll put that on. All right, now you should be able to see the screen. The lab is an integral part of the course. It is separate, but is a part of the course, and the grading of the 720 points is partly from the lab, obviously the lab projects, but the two big projects, the infectious disease project and the identification unknown, are towards the lab, or the points towards the lab. Come on. We will have some virtual labs in this lab. If the virtual lab uses Flash, you are not required to use it because Flash will no longer work. And I probably have the links crossed out. those links. But all virtual labs that don't use Flash should work, and you should use them, meaning complete them. Just a little note about the labs. All right, let's move down. You can read about safety information about COVID-19. I believe college is now making it optional if you wear a mask or not, if you stay 3 feet away from another student or not, and actually you should stay 6 feet away. And I don't know about vaccination. They may require students to be vaccinated still or to have a valid reason for an objection. Lab safety is important. So the safety rules are going to be briefly described. We go over this always in the first week. You're not physically going to be meeting in an actual lab, but we will go over the safety protocols as it's a component that's required for this course, and many nursing programs and other health allied programs require safety as one of their main skills that they want students to learn in microbiology. So never take shortcuts, which will risk your safety and your accuracy, as well as the safety of others in the lab. You are not allowed to take personal items into the lab and take them down to your lab bench. Personal items must remain in your book bag, and there is a space provided by the door for putting books, and those we call the cubby holes. And we actually consider the cubby hole and the door of the lab outside of the lab, and everything should be put in there. And then when you're done with that, you go wash your hands. So now you've entered the lab proper. And then you put on your lab coat, go find where your lab coat is hanging up, and then put on gloves, button up your lab coat, put on the gloves, and then you go to your workstation in the lab. So all of that, except for coming in the door and putting your books and coat in the cubby hole, is inside the lab. Is that making sense? So the cubby hole in the door we consider outside the lab, and you don't have to worry about keeping that area sterile, but everything in the rest of the lab, we don't want microbes to come into that lab, and we don't want microbes to go out of the lab or contaminate you. And that's why we require you to wash your hands before coming in, putting on a lab coat, putting on gloves. And then to be further safe, if ever you're working with a splash hazard, and that's usually a liquid culture containing bacteria or other microbes, you have to wear goggles. Or a splash hazard that would be something toxic to your eyes. You have to wear goggles. So all personal things should be in the cubby hole. If anything that's personal comes out of that region, meaning into the lab proper, it would be confiscated and would not be returned to you until it has been sterilized by either soaking in bleach for 15 minutes or being put into steam and pressure, what we call an autoclave, for 15 minutes. So if you were to take out your phone in the lab, it would be confiscated and not returned to you until it's been bleached or autoclaved, either of which procedures would destroy it. We provide materials for you to use in the lab, such as a writing pad, something to write with, which is usually a pen, a pencil, and then a marking pen. Things like tape, we provide that. Everything else should not come into the lab. And that's to prevent microbes from leaving the lab. Any question about that? And then at the end of the day, when you're all done, I haven't talked about your workspace, so I will talk about that in a minute. When you're all done, you take off your lab coat, actually take off your gloves first. And if they're not contaminated, throw them in the trash can. Take off your lab coat, go wash your hands, and then take out your books and your coat, and then you can leave. Any question about any of that procedure? Now, once you get into the lab, before you start, you should always wipe down your surface area where you will be working with disinfectant and a paper towel. And then before you leave, after you're all done and before you leave, you should also wipe down your work area with disinfectant and a paper towel. And that's to reduce the number of microbes that'll be in the lab, especially for the next person who comes in to work in the lab at your same workstation. All right, any question about any of that? You should consider all cultures that you're going to be working with. By a culture, I mean a microbe that's growing. You should consider it potentially pathogenic. You should keep your hands, pencils, and other materials out of your mouth, your ears, your nose. No eating or drinking, smoking, gum chewing, or any other oral habit is allowed in the lab. You should not touch your face, as I'm trying to demonstrate. Hopefully you can see that. I'm not even seeing that. Why am I not seeing that? Usually I see a little window. Anyways, you should not even touch your face while you're in your lab. But if you're the type of person who does that, you should just realize that you do and try to avoid it. And if you do, you just need to wash your face before you leave. You should wear proper attire in the lab. Besides the lab coat, you should wear closed-toed shoes. That's to provide minimal protection for your feet in case a microorganism is spilled on your feet. Okay, and as well as provide a little bit of protection for your feet in case something that's glass and is spilled and it breaks, and then we don't want you cut, but mostly for the microbes. Closed-toed shoes could be something like tennis shoes. Actually, solid leather shoes is best, but we don't require that. The point is you can't wear thongs or sandals or any of the women's fancy shoes that are open-toed. All right, you guys can probably see this. I need to save the lecture. It just finished. I think I want to save it here. Is that today? 4 or 5? Yeah, that's today. So I do want to save it there. All right. Normally that finishes before I start the lab, but we were a little late, and for some reason the computer was really slow. Or maybe they've changed the procedures and it'll just take more time. Cleanliness is a virtue in the lab, so always disinfect the lab bench before you start and then after you go. And then always wash your hands before you come in, or as soon as you come into the lab and before you go. Accidents should be reported to the instructor. Minor accidents should be cleaned by clean up broken glass first, but be very careful with broken glass because you can cut your skin and go through the gloves, break the glass. Use a dustpan and a broom to clean up broken glass. If you have a contaminated bacterial culture, clean it as the following. Cover the spilled material with lots of disinfectant and then put paper towel over it to wick the disinfectant everywhere. You can also put disinfectant over the paper towel, let it sit for 15 minutes before you wipe it up. If you have broken glass, put it in the broken glass container. The other, like the paper towel with the disinfectant and spilled liquid culture, put it in the autoclave bag or the biohazard bag. Biohazard bag is the same thing as autoclave bag. You should keep hair and paper away from Bunsen burners and back to incinerators. And then when you're not using a back to incinerator or a Bunsen burner, you should turn them off. You should never take a culture out of the lab. You should not bring cultures into the lab unless you get approval by the instructor. Old specimens should be discarded properly in the following way. Agar plates should be discarded in the appropriately labeled container. If it's a used autoclave plate, Petri dish with bacteria or other microbes in it, it should be put in the biohazard container. If it is a Petri dish that has not been used, and let's say it's broken or got contaminated to the air and you don't want to use it, it should be put in the trash can. Most agar plates would be put in the biohazard container. Glass slides, cotton swabs should be put in a glass container containing disinfectant. And we have a container for slides as well as cotton swabs. The cover slips to the slides are put in a separate glass container with disinfectant, and we do not reuse the cover slips. They will just be disinfected and then thrown away. But the slides will be cleaned and then, well, they're already disinfected because they're in disinfectant, and then reused. Test tube should be put, with all labeling tape removed, should be put in the test tube rack in the discard bin, and those will be autoclaved, washed, and then reused. Supplies needed for the lab will be found at the front of the lab. And then supplies for you to use in the lab, like I said, the paper, the pencil, will be in a drawer. And that drawer will be for your use, but it's going to be a shared drawer, meaning that students in another lab at another time can use that drawer. You are required to follow all of these rules. Failure to do so will risk your safety and the safety of others. And we'll talk about how you use a plate and how you use it to keep the bacteria in the plate and in your local environment so that it doesn't spread outside of the plate and into your workstation, and then from your workstation to other people. All right, any question about laboratory safety? All right, so on the schedule, you can find what we're doing in the week on the right side under the laboratory. Lab 00 on lab safety training is due 8 a.m. Friday, April 8th. And you will be dropped from the class if that is not submitted on time, or you do not email me and say that, ask me not to drop you from the class. Lab 01 will be due at the time most labs are due for the week, and it'll be due at 11:59 p.m. on Saturday. And most of the labs will be due, if not all of them, except for Lab 00, will be due on Saturday at 11:59 p.m. of the week we do that lab. Now, Lab 01, we will not talk about, and we will not do that until Thursday. You should read the infectious disease research project because I'm going to talk about that next week. So you should read about that. You can find that under Canvas in a file called Project Instructions. And I believe that's under the lab week 1 or lab week 2. You should also note that the online plagiarism quiz is due 11:59 p.m. this Saturday, April 9th. You must achieve 100% on the plagiarism quiz. So don't wait till 11:50 to get it done, because if you don't get 100%, you'll need to take it again. And if you don't get 100%, you'll get 0 on a 100-point project, and it's the infectious disease project. And we'll talk about that in week 2. All right, any questions about what we're doing today? If not, I'll move on. Talk about Lab 00. Can somebody confirm that I have you, just because I don't see anything?
Yes.
We're
Okay,
here. Yeah,
great.
Yep, I see it.
I can see the lab worksheet number 0.
Yes,
Yep, I
lab
can
work
see it.
0 is what we're going to work on. I'm going to tell you that if you use this online, page 1 will look like this, but if you print it out, it'll actually have questions on it under here. And I don't know what's going on there. I don't care. So I said not to mess with it. Come on, mouse. And so on page 2, I've got the questions. So you can ignore the above. And here's the lab safety word scramble. You need to fill in each asterisk for each question. Rule 1 in the lab, what do you do to wear? What do you wear for safety before you enter the lab? And I'm missing something here. Where's lab 1? Just a minute. I'm supposed to have the lab module, and I only have the worksheet. I might need to go online because it looks like I might not have it here. Oh, there it is. So go ahead and read through lab module 00. And if you come to the lab page, so I don't need that anymore. So let me close that down. There's the lab module, and there is the worksheet. And let me open that just to make sure that works. Looks like it's working. Yep, it's working. All right. So let me talk a little bit about the lab module, and then we'll talk about the worksheet. So you must work in a safe manner. Online students are not going to work in the lab, but you do need to know this material, as many of these rules will be relevant during your career, and you will be tested on these rules. So know the learning objectives, be able to distinguish between safe and unsafe laboratory practices, understand where the relevant lab safety equipment is located in the lab. And I won't really quiz you on that because you're not going to go into the lab, but understand how to safely dispose of contaminated materials. So you must wear a proper attire. I've already talked about that. Follow the safety rules. Wear a disposable glove, closed-toed shoes. And if you have a splash hazard, you have to wear goggles. If you don't have a splash hazard, you don't have to wear goggles. But if ever you're working with microorganisms in a liquid culture, you have to wear goggles. And if you're ever working with something that's hazardous to your eyes, like the colorizing agent in the gram stain, you have to wear goggles. Long hair should be tied back. I've already gone over this, so I'm not going to go over it again. This is what somebody should look like when they're in the lab, wearing gloves, a lab coat that's buttoned up, hair tied back. They're actually wearing a net, but we won't require that, just long hair tied. And you should have closed-toed shoes. I've already talked about that. That's actually a picture of our lab. You need to wash down the lab workspace you use, which would be here, before you begin, and then when you're done. And there they're doing that. All right, where you put waste products that you're trying to dispose of, regular trash that is not contaminated with bacteria, gloves that are not contaminated, and non-sharp objects that are not glass should be put in the regular trash can. In the biohazard container shown here should be any of the A that is contaminated, like your Petri dishes that have bacteria growing in them. When you're done with them, they should go in the biohazard thing. Any paper towel that you had that got bacteria on it should be put in the biohazard container. Even if you get disinfectant on the paper towel with bacteria, it should go in the biohazard container. If you spill bacteria on your gloves, the gloves should go in the biohazard container. Any question about any of that? Contaminated test tubes and bottles, which we almost never use, go in a different bin, which would be in the back of the lab. That's not stated here. And you put the test tubes in the test tube racks, the glass containers in the container for cleaning. Slides, we have a separate container for that, and it contains a disinfectant in it. Cover slips, we have a different container for them. And then soil, cotton swabs, we have a container for them too. The slides and the cover slip containers are found at the back of the lab. The cotton swab container can be at the back of the lab, but I think there's one at each bench table. And let me see if that's shown in the picture here. It's not shown in the picture there, but there would be, like on this table here, there'd be one container right here for this lab bench for putting cotton swabs in it. And that might be it there, but I can't see what that is. Broken glass goes in the broken glass container, and we have a container for that in the side of the lab. And you don't need to know where it is because you're not going to be in a real lab. Report all spills and accidents immediately. Cover microbes with paper towels and saturate the paper towels with disinfectant. Allow it to soak for 15 minutes. We allow it to soak for 15 minutes to kill all of the microorganisms. And then when you're done, pick this up, wipe it up, and then put it in the biohazard container. If there's broken glass, the broken glass goes in the broken glass container. We don't really use sharps. We do have a sharp container. I'm not going to go over that. You can read it. For using a Bactol incinerator or a Bunsen burner, you put your tool that you're working with, and this is a loop, and put it in touching the heat or in the flame, and then you make it red forever the wire portion of the loop. And you just look at it, and when it turns red, you pull it out. Usually it turns red in about 10 seconds or less, especially the flame. You only need to put it in the flame for a second or two. You should turn the Bunsen burner back to incinerator off when you're not using it. And that's just a safety precaution. And these are some of the safety equipment we have in the lab. A fire extinguisher, a fire hose, and a fire blanket, a fire alarm, a first aid kit, a deluge shower. So if you were ever to get something all over you and you wanted to take a shower in the lab, you just pull that down and it would shower you. An eye wash station. And I have to say, I've never had a student using, well, this would not be used in the lab, microbiology lab, unless you were to somehow splash acid all over you. Nothing we have is really requiring a shower. And a shower isn't going to help you for getting microbes on you. If you get something in your eye, you could use an eye wash station. And luckily, I've never had to use that. That looks like another fire alarm. All right. We also have a fire blanket, which I mentioned was there, and that you wrap around somebody who's on fire to smother the fire. I suppose if you ever caught on fire, you could also go underneath the shower. All right, so this lab, final 2 pages, detail all the safety rules that apply to a microbiology lab at Clark College. You're expected to follow and know these rules because you will be tested on them, as well as it'll be helpful to you in your career, assuming you're going into the health profession. Safety is of primary importance in the lab. All personal items should be left in your book bag or your purse or your coat, and then put in the cubby hole. No personal items should go to the rest of the lab, which is what we call the lab. The cubby hole we consider, and the door outside the lab. All right, I've already talked about that. Talked about that. Talked about cell phones. Talked about that. You should monitor yourself for a possible infection with the bacteria you're working with, such as getting diarrhea, fever, abdominal cramps. Obviously, you can't do that because you won't be exposed to live bacteria in an online class. But we have a document about the safety information online, detailing all the safety information you should follow, and then information you should take to a physician if ever you need to, which should never happen in an online class. I already talked about all of this stuff. Oh, here's something I haven't talked about. If you have a cut or a sore on your hand, you should wash your hand and then go and get a Band-Aid from the first aid kit. Put the Band-Aid over the cut and then put the glove on top of the Band-Aid. Okay. And then when you leave the lab, remove your glove, wash your hands. It's up to you what you want to do with the Band-Aid. You can leave it on or remove it.
I have a question about that,
Uh-huh.
actually. So, like, say this lab was in person this quarter. My hands get really dry because I have to wash my hands like all the time for work, and they're constantly open. What would you do in that scenario? Would I like just bandage my whole hand? Or
You're going to be wearing gloves in our lab. So regardless
Yeah.
of how many times you wash your hands, you'll be wearing gloves. So they shouldn't dry out. You will have to wash your hands at the start and the beginning. And if you need a Band-Aid, put it on. Now, what you do for your whole hand being cracked, I'm not sure. I've never had that happen. You could apply many Band-Aids. You could cotton swab your hand. And I would say do that before you come to the lab. And gosh, what would you do? Because you need to wash your hands and then cotton swab your hand. So I'm not sure what you would do in a case like that. The best thing maybe to do would be to take the class online for a week and then go back to and wait until your hands get better. Okay. Because yeah, if you wanted to Band-Aid your whole hand, you're going to require a lot of Band-Aids.
Yeah.
Okay. And I don't know if we have cotton swab. I've never looked for it in the first aid kit. They might have it, they might not. But you can't really cotton swab before you come to the lab. You have to bring the cotton swab with you, wash your hands, and then put on the cotton swab. And that would be okay. And then put on the glove. Okay.
Okay.
All right. We talked about discarding specimens. I think we're done talking about the lab module. Any questions about anything else? No,
Yeah,
no.
in the lab exercise that we have due at the end of this week, it lists the the safety data sheets to look over and review. Is that just something you want to look at online, or is that something you want to go over in the
Talking
course?
about rule 3, I haven't gotten to that yet, but yes, we talked about lab rule 1. What do you always wear for safety? We went over most of that, but please note, you will need to put 4 things. Okay. And this is before you enter the lab. So don't put gloves and a lab coat, because you don't put those on before you come to the lab. You put those on after you come to the lab. And there's 2 things you do for rule 2. After you enter the lab, what do you put on to keep yourself safe in the lab? Okay, so what do you put on to keep yourself safe before you enter the lab? And I'll give you a hint. You're going to be wearing clothes, okay? And that would be largely for rule 1. Rule 3, you have to look up safety data sheet online, meaning it's not in the lab module. Generally, all questions in the lab worksheet come from the lab module, but Roberto Anatori decided that there's nothing in the lab, the safety data sheet is not mentioned in the lab module, and he wants it because he thinks it's important for you to know. And so you have to look up what is the safety data sheet online, and then it covers different chemicals. And for that chemical, what does the safety data sheet or SDS describe? And you have to have 1, 2, 3, 4, 5 items. So make sure you fill in each asterisk. You leave one blank, you got one wrong. All right, know the location of your safety equipment, and you just need to name the safety equipment, such as a fire extinguisher. I'm giving you one answer, and then put in 5 different answers. And what can you take into the lab? And you can write no personal items, but in reality, you can take in some personal items. Like if you have jewelry on your ears, we don't require you to remove that. So that can be something that can be taken into the lab. We don't require you to take off your personal glasses before coming into the lab. So that could be something. And then the last one, can you eat and drink in the lab? And that's just yes or no. And then 2, list one item of laboratory attire you must wear in the microbiology lab. And that can repeat question 2 or 1 above. And then list 3, list one item that's allowed in the microbiology lab. And then list one item that's not allowed in the microbiology lab. and then submit this by 8 a.m. Friday of week 1 in the folder worksheet lab 00, which is right here. And you'll have a link for submitting it. Gosh, why am I not seeing? Oh, this is the module. So it should be right here, off on the top right, right there. You'll have an upload link. I got the download and the speed reader link. Okay, any questions?
So are we not doing the top half of that worksheet? We're just doing the questions, correct?
The top half is the questions. It's just that you can't see the questions until you print it out.
Okay.
Okay,
Got it.
and no, you don't have to do the top half of the worksheet. Okay, so I'll be here to answer any questions. And I'll be here until 8 o'clock. So get started on the lab. You can work on the lab while I'm here. And you don't have to, you can work on the lab anytime before 8 a.m. Friday. Okay. All right, any questions?
If you don't have a printer at home, how are you supposed to see the questions?
I've got the questions on page 2. Like I said, you can ignore page 1. Okay. All right. I'm assuming everything's fine. I'm sorry I can't see anyone, so I don't know what's going on. Let me see if I can bring this here. I can stop sharing now and then I'll start seeing you. Okay, great. All right. So those of you who want to work on it, I will be here to answer any questions till 8 o'clock. At 8 o'clock, I will log off. And if a student, all students log off before 8 o'clock, I will probably log off also. So answer questions beforehand. All right,
Hi,
go ahead and
Dr.
get
Vol.
started.
Is someone else speaking? I'm sorry. Or was it just me?
I think it's just you.
Okay, so when I'm looking at number 1, in the lab, what do you always wear for safety before you enter? What is the 4th thing? Because it's kind of, the question is contradicting. So it says, in the lab, what do you always wear for safety before you enter the lab? So I've got closed toe shoes, hair pulled back, clothes.
Okay, well, you're being very general with clothes. You can say
Oh, okay,
shirt and slacks.
gotcha. Okay, I didn't know that we could be
Yeah.
that generic.
Yeah,
Thank you.
and there's other items too, but those are the main ones. All right.
So I have a question about lab coats. Just wondering, why do we use lab coats instead of the isolation gowns that we use in hospitals?
Well, an isolation gown, although they're disposable, and they'd be better, we use lab coats so they can be washed and then reused. Meaning what we do is most students use the lab coats we have in the lab.
Mm-hmm.
And then at the end of the term, those will be washed, cleaned, autoclaved, actually autoclaved and washed, and then made available for the next term. The disposable ones, the safety ones, you can't autoclave them. And I don't think you can wash them either. I don't really know.
No, you can't. You have, it's, they're single use.
Yeah. So that's
So-
why.
Yeah. So just wondering, so there's no worry about transferring any microbes from the lab coat onto you as you're putting it on or taking it off?
If you were to have a major liquid spill, it could contaminate you, but a minor spill, even if it's liquid, it shouldn't go through the lab coat. Okay.
Okay.
And so that's what we're doing. If it's a major spill, I've never had that, but if you got it on yourself, you should disinfect the area that's wet underneath your lab coat.
Interesting, thank you. Yeah, I'm just interested in like infection control because I work in a healthcare setting and we always do things a certain way to minimize the spread of infection. And so it's interesting to see how what we do at work differs from how we might do it in the lab. So
Yeah,
thank-
I think it's mainly that they decided not to provide, I mean, a disposable one, you'd have to reuse it all during the lab, and if it gets a tear in it, it wouldn't be effective. So I think they decided to go with lab coats, and then they only really need to buy them one time, and then they can continue using them because they've used those lab coats for at least 10 years now.
That's true.
Yeah. So I think that it was mainly cost and effectiveness and how they can supply it.
Okay,
Well,
that
that's
makes sense.
Yeah.
Thank you.
All right.
I also have a question. I just tried printing the lab 00 and all I got was, I did not get the pictures, like the questions on the pictures. I just got the lab safety module 00.
Interesting. You're the first student who's told me when they printed it, they didn't get the questions, but that's okay.
I mean, I
Um,
got the
You got the
question.
questions on page 2. Just answer those questions.
Okay, thank you.
All right. Oh, we can stop the recording.